Bismillahirrahmanirrahim. Welcome back, friends. Welcome to another video lecture by Kami Biology. So we talked about the introduction and we talked about the different types of bacteria according to the flagella. Now this is the part B of the flagella, and in this video lecture we are going to talk about structure of the flagella. So let me write structure of flagella. Okay. So now if we see the structure of the flagella, so the structure of the flagella, first of all, it can be studied by transmission electron microscope. This is the first thing. The second thing is that the bacterial flagella structure is very complicated and a very complex structure. Now it consists of three main portions. The first portion we call that, let me write the name. So the first portion we call that Fallament. The second portion we call that hook body, and the third portion we call that what? Basal body. Basal body or basal portion. Now to explain each of the portion of the flagella means this third one, basal body, is very complex and complicated. What actually happened? So what actually happened? Let's suppose this is let's suppose this. Actually, these flagella are present in case of gram-positive bacteria and in case of gram-negative bacteria. Okay, inshallah, I will also discuss the flagella structure in case of the gram-positive bacteria as well. But here, actually, I'm actually talking about the flagella structure in case of the gram-negative bacteria because it is very interesting in gram-negative bacteria. Now, what actually happened in case of gram-negative bacteria? This is, let's suppose, the inner membrane. Okay, because all of you better know that gram-negative bacteria have two membrane. One we call that outer and the other we call that inner membrane. Now this is the inner membrane, and this is what? This is the let's suppose the peptidoglycan layer. Okay, and after that we have the outer membrane. Okay, so this is what? This is then the outer membrane. Okay, so first of all, let's start from the basal body. So they have three portions, means this basal body have three portions. The first we call that rotor, the second we call that stator, and the third we call that bushing area. What actually happened? First of all, let's start from the rotor portion. So the rotor portion is actually consists of different kinds of the proteins, fly proteins, and it consists of different kinds of the rings. So let's suppose what actually happened. So this means this rotor area consists of different fly proteins. So the first protein we call that Fli F protein. This Fli F protein can make a ring. We call that M and S ring. So the first we call that, so let me draw the ring. So first of all, what we have a ring which is actually synthesized by what? Which is actually synthesized by the Fli F protein. So let me write Fli F protein. So let me write here. So this is what? This is the Fli F protein. Now this Fli F protein can make two rings. So this is the first ring, and second we have another ring. So let me draw here. So this is the second ring. The first ring we call that M ring, and the second ring we call that S ring. Okay, so we have two ring, M ring, and this is the S ring. Okay, so this Fli protein are actually responsible to make M and S ring. After that we have another proteins, we call that Fli M and Fli N protein, which can make the rest of the rotor portion. So after that, what we have another proteins, we call it Fli G proteins. So Fli G proteins are actually present below the S ring. So this is what? This is the Fli G proteins. Okay, so we call that Fli G protein. Now Fli G protein will be present below this S ring. After that, once the Fli G proteins are formed, so then we have another portion of the rotor, or we can say we have another portion of the rotor. So what we call that? We call that means this portion of the rotor is actually made up of we can say Fli M and Fli N proteins. So this is another portion. Okay. Now it is made up of what? It is made up of Fli M and Fli N, guys. So all this portion, as you are watching, all this portion are called, means this Fli F, S, M ring, Fli G, Fli M, Fli N, all this portion are called what? All this portion we call that rotor. First thing. The second thing is about that rotor is actually moving, or they are actually in a circulating form, means they are circular in form, first thing, and they show a motion in a circular form. So this is the first thing. The second thing is about we have another protein. So let me a little bit draw more clearly it. Okay, the second we have stator. So now stator is actually, it is a silent, and we also say that it is actually a stationary form. Means this stator is actually in a stationary form. What actually happens? Stator area will be present upper area of this Fli F protein. Now second thing that I want to tell you, this Fli F protein, or we can say M ring, or this S ring, means this M ring will be connected with this inner cell membrane, while this S ring will be connected with some of the inner membrane area and with the cytoplasm as well. The second portion, this Fli G protein, will be present in the cytoplasm area. Similarly, this Fli M and Fli N area is also present at the cytoplasm area. Okay, then we have another portion of the basal body, we call that stator. So stator are usually in a stationary form. So what actually happened? This is a stator, means this stator again consists two portion. Okay, so it is actually made up of two protein, we can also say that. So this green one portion, as I'm drawing, this is the first portion, which is made up of first protein, and this is the second one portion. Okay. Okay, so this one portion, as I draw, means this bottom area as I draw, we call that Mot B, and this elongated portion we call that Mot A. Okay, so these are the stationary area, and it will be fixed, first thing. Second thing is that it is not in a motion form, it is in the stationary form. So what actually happened? This we call that what? We call it stationary, stator. So once these portion are formed, means once this basal body are formed, so then what we have another portion of the basal body, we call that filament proteins. So let me draw here. So then we have a filament proteins formed. So these blue one are what? These blue one are the filament proteins. The first thing that you should have to keep in mind, filament, inshallah I will discuss the synthesis of the flagella, that how actually the flagella are synthesized. I will discuss it in a detail. But what actually happened, let me tell you a little bit here. There are a messenger RNA, and we have messenger RNA and ribosomes. Ribosomes and messenger RNA are connected together, and they make a protein, we call it filament proteins. Okay, so the filament proteins are connected, and they make a rod here at somewhere, as I'm drawing. So they make a rod here. So this is actually in a rod form, and it is, first thing, it is a hollow. Okay, as I am drawing very complex and complicated, but actually it is a hollow. The first thing. The second thing is that after this hollow tubes are formed, or we can say rod are formed, so then they will be connected another two rings. So let me draw. So they will be connected another two rings. Okay. So the first ring we call that P, ring, and the second we call that L ring. L ring. Now once P and L ring are formed, so then what actually happen? What actually happen? There are a hook caps are formed. So this one area, as I drawing this one area, are called what? Hook cap. Okay, what actually happen? Hook cap can make we can say that hook cap can make a hook portion. So this basal portion are now end. Then second we have a hook portion. So what actually happen? This hook portion can make a hook body. Okay, so this is what? This is the hook body. So this is the hook portion. Okay, they are usually in a means this, they are a little bit turn, means this is a little bit hook in shape, as you are watching. Now these hook caps are actually connected. When the hook caps are connected and when the hook caps moves, so with this movement of the hook caps, they can make what? They can make hook proteins and they can make hook portion. Let me tell you a little bit. So let's suppose if this is the hook cap, so with the help of this hook cap, the filament proteins will be connected. The filament proteins will be connected and they can make the hook portion. When the hook cap is not present, so it is impossible, or we can say it is not possible for the filament proteins attaches. So the filament proteins attachment assembly and they are added with each other and they make the structure of the hook. It is possible by this hook cap. What actually happen? This hook cap moving in the upper direction. So when the hook cap is moving in the upper direction slowly, so with this direction, upper direction, so then the proteins, means the filament proteins, are connected and added with each other and they make the hook portion. Okay, after that what actually happen? This hook caps, let's suppose if this is the hook cap, let's suppose this hook caps will be means removed out. So this is the hook cap, so they will be removed out. After that we have a filament cap proteins. So here we have what? We have another proteins, we call that filament cap proteins. Filament cap proteins. Now this filament cap proteins are also do the same things. What actually happen? This means this they will produce what? They will produce a filament. So finally what we have? We have a filament. Now this is the filament portion. Again the filament are also synthesized when the filament cap proteins are present. The filament cap protein, just like, again let me tell you, explain again what actually happen. If this is the cap proteins, means this is the filament cap proteins. So when the filament cap protein are moving in the upward direction, so then they will means connect, or they will produce the filament proteins, or we can say the filament proteins are connected and binding and assembly with each other and they make what? They can make this filament portion as well. Okay, so this is what? This is the filament portion. So this is the filament portion. Now one thing that I forget to mention that this is about the rod. Now this is what? This is the rod portion. Okay, and another thing that I forget to mention, this M and this PL ring are connected to each other and with this rod portion. Mean this rod portion P and L, we are collectively called that what? We collectively call it bushing. Bushing. Okay, so as I told you, this that this basal body is actually very complicated. Why? Because it is connected with this rotor portion, stator portion and bushing portion. Okay, and this rotor portion, as I told you, it is made up of MS rings and it is also made up of fly proteins as well. After that, then we have Mot A, Mot B proteins connected to each other and we call it stator. And then after that we have a bushing portion, means P and L ring are connected. With this PL ring we have the hook area and then we have the filament. Now the second thing is that this hook portion and this means this basal portion, these two portions are actually slightly wider. Okay, as you are watching, this hook and this basal portion are wider. That means, and similarly, if we see, this filament portion is actually not wider, it is thin. Okay, moreover, this filament, if we see the filament and if we see this hook and filament portion have an extra layer. So let me draw this extra layer with this black marker. So they have an extra layer, as I draw here. So they have an extra layer which is made up of lipopolysaccharide. Okay, so actually this flagella, if they are present outside, means arises from the outside, so they have an extra layer, an extra sheeted, we usually called it lipopolysaccharide. Now as you are watching, this P and L ring are usually connected with this peptidoglycan layer, and they are usually connected with this outer membrane area as well. And this hook portion are usually connected with this outer membrane portion. And if we see, this filament area are always connected with this hook portion as well. So okay guys, so that's all about the structure of the flagella. So it is again, let me tell you, it is very complex and complicated, but I explained it very simply. Okay, but if still you have confusion, or if still you have some questions in your mind, so write it into a comment. Inshallah, I will give you answer as soon as possible. So I hope this videos help you. Inshallah, in the next video lecture of this flagella, we are going to talk about the synthesis of the flagella, that how actually the flagella are synthesized. And after that, then we are going to talk about the flagellar movement, that how actually the flagellar move and what direction they are move. Okay, so stay tuned and continue to watch the rest of the video. Thank you so much.